Welcome to Lecto. Today we are going to discuss vitamin B12, which is also called cobalamin. In this lecture, we will discuss the structure, distribution, different reactions and the absorptions of the cobalamin. So let's start. First of all, we will discuss the important components of the cobalamin. Cobalamin consists of the corrin ring, and the corrin ring consists of 4 pyrrole ring. So if you see here, this is the corrin ring and this corrin ring consists of 4 pyrrole ring. So here you can see these are basically the pyrrole ring. So and you see that this is the one pyrrole ring 1, 2, 3 and 4. Now if you see that each pyrrole ring consists of the nitrogen, and that nitrogen will attach to the central atom and which is the cobalt. So the nitrogen of the pyrrole ring attached to the cobalt, and in that situation cobalt form 4 bonds. Okay, but the cobalt has ability to form 6 bonds. In that case you will see cobalt form 2 other bonds at upside and at the lower side. At the lower side, cobalt form a bond with the substance 5,6-dimethyl benzimidazole. So this is the compound 5,6-dimethyl benzimidazole, and at the upside it has ability to attach with the 3 different compounds. So if you see here that in this case it can either attach to the methyl group, okay, cyanide group and 5' deoxyadenosyl. So one of these 3 compounds can attach with the upper side. So in that situation, if you see here, if the methyl group is attached, then it is called Methyl-cobalamin. If the cyanide group attached to the upper part, then it is called the Cyano-cobalamin. And if the 5-deoxyadenosyl attached, 5' deoxyadenosyl attached, then it is called 5' deoxyadenosyl cobalamin. So this is the situation. But the important thing that is that cyanide form is actually the synthetic form. This is the synthetic form. But the methyl and the 5' deoxyadenosyl is actually the coenzyme form. So these are basically methyl, and the 5-deoxyadenosyl is actually the coenzyme form. These are basically the difference between these 2 forms of the cobalamin. Next we will discuss the distribution. Actually cobalamin is formed in the microorganism and it is not present in the plant. This is the situation that the cobalamin is actually formed by the microorganisms, and you can obtain cobalamin only from the animal like liver, fish, meat. So these are basically the different animal sources: liver, meat, fish. So these are basically you can obtain cobalamin only from the animal sources. So this is the distribution of the cobalamin. Next we will discuss the important reactions of the cobalamin, how cobalamin involved in the conversion of the different substances. First of all, we will discuss how methionine formed with the help of the cobalamin. So if you see here that homocysteine convert into the methionine. Methionine. This is the important point. And methionine is actually the amino acid. And in that system, you will see this conversion is happened with the help of the enzyme, and that enzyme is actually the methionine synthase. Okay, good. Now, as we know that the enzyme required the cofactor for its proper functioning. So, methionine synthase required the cofactor, and that cofactor is the cobalamin. What forms of cobalamin? Actually, two forms of the cofactor cobalamin exist. One is the methyl-cobalamin and second is the 5-deoxyadenosyl cobalamin. So in that system, you will see the methyl-cobalamin. This is the cofactor form of the cobalamin. Work as a coenzyme with the methionine synthase. Okay. And in this system, you will see the addition of the methyl group to the homocysteine, then it will convert into the methionine. If you see here, and this happens with the help of the N5-methyl tetrahydrofolate. If you see my previous lecture in which we discussed the folic acid. So N5-methyl tetrahydrofolate transfer the methyl to the homocysteine, then it convert into the methionine, and after that you will see it will convert into the tetrahydrofolate. Okay. Now again, in that system you will see tetrahydrofolate convert again into the N5-methyl tetrahydrofolate. Okay. But there is a situation, there is a theory in that system which is called the folate trap theory. In the folate trap theory, you will see if the cobalamin is not present, then what will happen? Then the conversion of the homocysteine to methionine will stop. Okay. And you also see the N5-methyl tetrahydrofolate cannot convert into the tetrahydrofolate. So in this system, you will see the tetrahydrofolate will trap in the form of the N5-methyl tetrahydrofolate. So with the passage of time, that concentration or the amounts of the tetrahydrofolate decrease in the body. But you will see the increase in the concentration of this form of the tetrahydrofolate. And that form actually trap the tetrahydrofolate. So this is the folate trap theory. Okay. Now next is and methionine is actually the amino acid, and that amino acid is actually used in the formation of the other compounds like the glutathione. So and SAM, S-adenosyl methionine, that is actually used in the repairing of the DNA. So this is the uses of the methionine. Second is the Succinyl CoA synthesis. So in that system, you will see here old carbon fatty acid plus amino acid convert into the methyl malonyl CoA. So old fatty acid and amino acid convert into the methyl malonyl CoA, and methyl malonyl CoA convert into the succinyl CoA, and this conversion is actually happens in the presence of the methyl malonyl mutase. This enzyme actually convert the methyl malonyl CoA to the succinyl CoA. Okay. And this enzyme also required the coenzyme or cofactor, and this actually 5-deoxyadenosyl cobalamin act as a coenzyme and attach to the butase. So what forms of the cobalamin act as a coenzyme? So you see 5 prime deoxyadenosyl cobalamin act as a cofactor in that situation, and you will see the formation of the Succinyl CoA. And this is important, as we know that the Succinyl CoA is important in the formation of the heme of the hemoglobin, heme synthesis. So this is important point. Next we will discuss the absorption. Before we discuss the absorptions of the cobalamin, we should know the transporters of the cobalamin. So actually you will see that the cobalamin is transported by the protein, and that is actually called the transcobalamin. Transcobalamin. They actually transport the cobalamin, and transcobalamin has two types: Transcobalamin 1, Transcobalamin 2. Transcobalamin 1, also called Hepto-corrin, or you can say R protein, or you can say Cobalophilin. So these are basically the different names of the transcobalamin 1: Hepto-corrin, R protein, and Cobalophilin. These are basically they transport the cobalamin in the different situation. So if you see here, like this is a person, and this person take a meal, and this meal has cobalamin. This meal has cobalamin. Okay, if this in the mouth, you will see the productions of the saliva. When this saliva produce, in this saliva there is a transcobalamin 1. So here if you see that this is the transcobalamin 1. Okay, now the food along with this saliva move into the esophagus and ultimately reach in the stomach. So if you see here, like this is the food, and this food contains the cobalamin. Okay, and here you see the transcobalamin 1, which is actually present in the saliva. Okay, now in the stomach you will see the parietal cell or axontic cell. So this is the parietal cell, or you can say axontic cell, that produce two things. First one, these cell produce the hydrochloric acid, and second is the intrinsic factor. Two things produced by the parietal cell. Okay, now if you see here, when the HCl produce, then what will happen? It actually dissociate the cobalamin from the food. So if you see here, now the food is separate from the cobalamin in the acidic environment. So now the cobalamin that is secreted from the food or separate from the food is now vulnerable to the hydrochloric acid. It can be destroyed by the hydrochloric acid. So in that situation, the transcobalamin 1 that is present in the stomach surround this cobalamin. Now this is safe. The cobalamin surrounded by the transcobalamin is safe from the hydrochloric acid. Okay, and if you see here, like this is actually the intrinsic factor. Two things produced from the parietal cell. Okay, now these two things move downward. Cobalamin surrounded by the transcobalamin 1 and the intrinsic factor. Okay, and as we know, at the start of the duodenum, we see the opening of the pancreatic. We see the pancreatic opening, and pancreas start its secretion into the duodenum. So in this case, you will see like transcobalamin 1 surrounded by the cobalamin. Now, this transcobalamin 1 break down by the pancreatic enzyme and which is called the protease. Now, in that situation, you see the protease enzyme break down this transcobalamin 1. So in that system, you will see the cobalamin is now free. And as we know, the intrinsic factor move also along with the food. Now if you see two things, intrinsic factor and cobalamin. So this is the intrinsic factor. Okay. Now the intrinsic factor will attach to the cobalamin. Now this will move and reach at the ileum. In the ileum, you will see a receptor. So, and this receptor is actually called the cubilin. This cubilin receptor attach the intrinsic factor along with the cobalamin. Okay. Now, this cobalamin will move through the enterocyte and finally reach into the blood circulation. So here is the cobalamin. This is the cobalamin. Now this cobalamin will transport towards the liver. So this is actually the liver, and in that system you will see a transporter, and that transporter is called Transcobalamin II. Now, Transcobalamin II will transport the cobalamin in the blood circulation and it will move towards the liver. And in the liver, transcobalamin will store, and approximately 2 to 5 milligram cobalamin is stored in the liver. So, this is the normal situation. And if you see that in what situation vitamin B12 or cobalamin will not absorb. So if you see that two situation in the autoimmune diseases and in gastrectomy. So these are basically the two situation. In the partial or complete gastrectomy, intrinsic factor will not produce. And if there is no intrinsic factor, then the absorption of the cobalamin will not be happened. So this is the case 1. And in the case 2, in the autoimmune, actually antibodies will attack to the parietal cell and destroy the parietal cell. So actually, as we know that the intrinsic factor produced from the parietal cell, if the parietal cell destroyed, then there is no intrinsic factor and there is no absorptions of the cobalamin. So in that situation, when the cobalamin is not absorbed, you will see the pernicious anemia. Pernicious anemia. When you see the cobalamin deficiency. So this is all about the vitamin B12, which is also called cobalamin. If you have any question, then you may ask in the comment section. Thank you.